Hello everyone, I am Zhang Jia. I am pleased to share with you our work on Raman flow cytometry-based single-cell species classification, viable cell counting and vitality test for probiotic products. Currently, there are significant limitations in traditional probiotic quality control methods. Its single colony detection accuracy, counting, strain identification and metabolic viability detection methods are limited in application due to defects such as reliance on prior pure culture, long detection period, and difficulty in handling composite products. The miRS technology developed by us can effectively solve these problems. It does not require pre-culture, quickly identifies strains through Raman signal fingerprinting, and determines cell death and in situ metabolic viability by using CD peaks in the silent phase, and the whole QC takes only 4 to 5 hours. Although miRS can solve the bottleneck problem of traditional methods, it also has the problem of low throughput. It requires repeated centrifugation, washing, and additional microscopic counting to calculate the total number of bacteria, and it takes 10 seconds to collect Raman spectra of individual cells, which makes the detection efficiency difficult to meet the actual demand. For this reason, we have developed the RFC technology, which can achieve direct detection without centrifugation, and the cells are queued at high speed, and it takes only 1 second to collect one cell. Through a single collection, the identification of strains, counting of viable bacteria and evaluation of probiotic viability can be completed. Compared, the process is simplified from 3 steps to 1 step and the throughput is increased by more than 10 times, which results in higher accuracy and detection limits. It was found that the average accuracy of the linear discriminant analysis classifier was 99.3%, which was better than the other 5 algorithms. When the number of collected spectra was greater than 1500, the prediction accuracy reached 99.26%. The average recognition accuracy between batches was as high as 98%, confirming that integrating multiple batches of data can effectively improve the accuracy. To evaluate the accuracy of the LDA model for strain identification in complex microbial communities, a simulated probiotic community consisting of BB12 and RHLP08 was constructed in this study. For each scale sample, the proportions of the 2 bacteria could be accurately predicted, and the deviation of the predicted values from the actual value less than 3.7% at each scale. When the relative abundance of BB12 was as low as 1:1000, the RFC could still achieve accurate differentiation by increasing the sampling depth to 5000 entries. We introduced a known quantity of a reference strain and mixed it with the test strains at a specific ratio. By applying the species identification method illustrated in this study, we were able to determine the relative ratios of the 2 strains. This, in turn, enabled us to calculate the absolute quantity of the test strains based on the known quantity of the reference strain. The results showed that the probiotic products could be accurately detected in the range of 1E6-1E12 cells/G with high linear correlation. The total bacterial counts of the 3 single-strain probiotic powder products detected using RFC were not significantly different from those of the conventional method. We used MAL as a criterion for viability determination. When MAL is greater than 0, it is considered to be a living cell, and when MAL is less than or equal to 0, it is considered to be a dead cell, and based on the value of MAL, the metabolic viability and heterogeneity indices of the products can be obtained. Through the parallel validation of the RSA and the traditional method for the 3 single-strain probiotic products, the results showed that there was no significant difference between the results of viability and viable bacterial count and the traditional method. BKE3 products contain Lactobacillus paracasei K56, AT22, and Bifidobacterium animalis BL99, and a reference database of single-cell Raman spectroscopy of the above 3 probiotics was constructed, and the experimental results showed the internal reference calibration method for total cell counting, and the results indicated that no significant difference with the traditional method, and the RFC could realize the identification of the strains at the level of the strains, whereas the traditional sequencing method was unable to distinguish between K56 and ET22. Based on the experimental results of strain identification, the metabolic viability and heterogeneity index of each strain BKE3 product can be obtained. Therefore, RFC provides a new solution for the quality assessment of probiotics and viable cell products with the 3 major advantages of high throughput, accurate precision, and multi-indicator integration.